So we are going to begin today by talking about the different types of microscopes. We'll just sort of give an overview about the parts of compound microscopes. And then this week we will get some practice using the compound and the dissecting microscope. And we'll work on a microscope lab. And then we'll start to move into cells once we understand how to use microscopes and so forth. So here's our unit. Some vocab that will come more into play sort of at the middle and end of the unit. So, microscopes. I know you're familiar with microscopes. You guys ever use microscopes in elementary school?
No.
All right, so microscopes are tools. They look like these are the types we will use in class. And there's two different types here. I'm going to use this to sort of show you how they work and how they work differently. This type of microscope that you see here, this is called a compound light microscope. This is the microscope we'll be using most often. And microscopes allow for the magnification of an image, so you can see things that are very small. And different types of microscopes are able to magnify an image different amounts. The compound light microscopes that we use are able to magnify an image from a minimum of 40 times to a maximum of 400 times. When you talk about magnification of a microscope, you're basically saying how many times larger does the object appear versus its actual size. So if you're using 400x magnification, it means what you see in the eyepiece looks 400 times bigger than the object actually is. The other type of microscope that we'll use a little bit is this microscope. It's called a dissecting microscope. And the dissecting microscope, that's the name, sometimes called a binocular microscope. It's not just used for dissecting, it's used for other purposes, which we'll talk about. It does not magnify nearly as much as the compound microscope. It goes from 10x to about 40x. Okay? Most of ours either go 10 to 20 or 10 to 30, depending on which microscope you get. The third type of microscope is one we don't have here. It's called an electron microscope. And electron microscopes, they look more like a computer than they do a microscope. They're not portable. You can't take them place to place. When you have an area with an electron microscope, it would take up about the size of this desk. You need that much area. Okay? It's got several pieces to it, a computer monitor, the area where you have the object and so forth. So we don't have one here at school. They can give magnification up to and even greater than 1 million times. So typically, you might see them here and there at schools, high schools. Whitesboro High School has an electron microscope. I was able to use it. They got a grant to purchase their electron microscope, so I went to like a workshop there, and we got to use it, and I brought an owl's feather, and this was the electron microscope image I took using the microscope. So we'll talk a little bit about the differences between the types of microscope here in a minute. So those are the three general types. So let's look at some images from each type, and I'll tell you a little bit about what makes these microscopes different. So with the compound microscope, the object that you're going to view goes on this flat part. It's called the stage. And in order to see an image, light comes from the light source down here, goes through a little glass sort of passageway, through the stage, through the slide that you're putting here, then through a lens here, bounces off a mirror here, gets directed through another lens up here, and then you can actually see the image. But in order to, do you think, am I going to be able to look at my hand under this microscope?
No.
No. Why not?
Too big for lights too.
Yeah, so the objects you look at in a compound light microscope, they have to be thin enough so that light can pass through them to continue the pathway to your eye. So you can't look at a solid object under a compound microscope. Typically when you look at objects in the microscope, they're mounted on little glass slides.
Yeah.
And so the objects on those slides are typically
I watched a YouTube video.
typically very, very thin slices of materials. For example, I have a whole bunch of examples of prepared slides. These are some slides that show human cardiac muscle.
Well,
Muscle from somebody's heart. And this tiny little, you could see it's like a pinkish purple stained section. That's a very, very thin section of muscle from a person's heart. And so you can't look at sort of three-dimensional objects. You can't look at thick, opaque objects. For that, you would need another type of microscope.
Okay.
So here are some examples of images with different types of compound light microscopes. This is an amoeba. It's a single-celled organism. Scientists first discovered that there are living organisms we can't even see living in, for example, pond water. And they learned this by looking at pond water under the microscope. They saw things like amoeba, paramecium, euglena. These are all single-celled living organisms. Vorticella, radiolarans, foraminiferans. These are all very, very small organisms. So when we look at the dissecting microscope, it's a little different. So first of all, the main difference that you probably notice is the eyepieces. What do you notice?
There's one and then there's two.
Yeah, there are two. So two eyepieces, and that results in a different image. Okay? Because you look through it with both eyes. You'll also notice the light is different. So if I put my hand on the dissecting microscope, the path that light follows is different than it is in the compound microscope. So if I put my hand here, where is the light coming from?
The top.
Above. It hits my hand, does what? Bounces off, and then goes through the lenses to my eye. So with a dissecting microscope, I can look at a thick, opaque, three-dimensional object. If I look through here, I'll see the surface of my hand in this dissecting microscope. So that's an important difference. The other important difference is the two eyepieces. So humans have, this is kind of a digression, but that's okay. Humans have two forward-facing eyes, as some other animals do as well. But some organisms have eyes that are not forward-facing, they're on the side of their head. Do you know what the advantage to each of those, how are each of those a helpful adaptation? What about eyes that are wide on the side of our head? How is that a helpful adaptation to some organisms? Jake?
Because if you're like standing here, you can see both ways.
Yes, it gives you a very, very wide peripheral vision. You could see, you know, I don't know the exact amount, 250 degrees around you. And so you could sense objects that are way on the side of you. Humans don't have peripheral vision that's as good as animals that have eyes on the side of their head. Okay, so what's the advantage to having forward-facing eyes? Ali?
You can see things better when they're in front of you.
Sort of. You're getting there. Ella?
This really helps you focus on like what's right in front of you.
Not quite.
Do like animals with eyes on the side of their head? Do they have like a blind spot?
No, not really. They could see 100% around them.
Do you have depth perception or something?
Yes. They give better depth perception. Do you know what depth perception actually means? Addis, what does it mean?
The ability to judge how far away an object is based on.
Yes, the ability to see how near or far an object is. Having forward-facing eyes improves that depth perception. If you think about like monkeys and apes, other primates, that sort of spend a lot of time swinging through trees in the forest. They have forward-facing eyes. It's really important to know where that next branch is as they're moving through the forest. And so, do you know why forward-facing eyes are better? All right, so I'll tell you why. I'm sure you probably have done this. If you hold your finger in front of like your right eye, for example, and open and close one eye and then the other, right? You've probably done this, right? What happens to your finger?
Can't see my finger. It's gone. My finger's
It moves. Is your finger actually moving?
No.
Why does it appear to move when you do this?
Your eyes are in different places.
Yeah, your eyes are not slightly offset. And so your right eye, right now my right eye sees my finger here, straight in front of my eye. However, my left eye is looking at it at a slight angle. And so when I switch right and left eye, my finger seems to move. So if you do the same thing, put it far away from you. How much does it move?
Not a lot.
Not as much. Look at an object across the room. How much does it seem to move?
Not at all.
Almost not at all. So what happens is your brain processes the image coming in each eye. And when your brain sees, each eye is seeing this at a very different angle, it tells you this object must be close, because each eye is seeing it at a very different place. If your brain notices both eyes are seeing the object in basically the same place, your brain says that object must be far away. And that's why forward-facing eyes give us good depth perception. Now, this is also, as a separate aside, how 3D movies work. Or did you ever see those 3D drawings that are in red and blue ink?
Yeah.
And they put on the red and blue glasses? What they're doing basically is one eye is seeing one image, your other eye is seeing a different image, and your brain puts those together. And so if the artist wants the object to appear close to you, they draw those two images far apart, the red and the blue ink. If they want the object to look like it's far away, they draw them right next to each other. And those glasses give each of our eyes a different view of the image. All right. Why am I going into all this? Because this microscope gives you binocular vision. You look through two eyepieces. Each eye is getting a separate view of the image, allowing your brain to get a sense of depth perception. When you look at these images, and when you use this microscope, you'll see the object looks three-dimensional. You can see the depth to it, unlike a flat image that you get from a compound microscope.
Yummy. Delicious.
Now, an electron microscope, there are a couple different types. There's scanning electron microscope
Don't
images,
like that. It's scary.
and there are transmission electron microscope image. A transmission electron microscope is more like this, compound microscope. Scanning electron microscopes are more like this. They give these three-dimensional views that you probably have seen.
I don't like that.
Like that.
I don't like that. Or that.
Is it possible for them to get like a constant?
So no. All right, a couple things.
Oh.
Oh, the pollen makes me have like trypophobia.
So these two images, or these different images, are all views from scanning electron microscopes. They give a three-dimensional view of an object. A couple things about using the electron microscope. Basically, the object you're going to look at has to be coated in a thin layer of metal. And therefore, you can't usually look at living things in the electron microscope. They have to be dead. So all these insects you see are no longer living. Also gives you a black and white image. An electron microscope doesn't use light to create the image. It uses electrons. It sends a beam of electrons at the object. They reflect from the metal and are picked up by a sensor. Because electrons are very, very tiny, as we learned, they give a very great deal of magnification and resolution. So when you see these color images, they've been artificially colored. They've made, the background green and the other parts red, just to allow you to have an easier time seeing them.
Yo. Ah. Ooh, that looks delicious.
It looks so fake.
Yes, these are what these insects really look like.
That doesn't seem
That looks delicious.
Look, it looks
Yeah, they
like
don't.
that.
So
I don't like
I
that.
said
Oh.
I said they give you an image a million times greater. They
Oh.
can. They don't have to. So when you're going to be taking the picture with the electron microscope, you can magnify only 10 or 100 times if you want to, but
It's
you
up to
could
that number.
go up to 100,000.
Because I'm like, I feel like you could get way closer
Yeah,
than that. I was
you
like,
could.
that must be the
It's just to
tiniest
show
bug
these.
there has ever been.
Oh.
Picture.
I don't like
All
that.
right, shh. Yeah, they artificially color them to make different parts like stand out. All right, so let's talk about the parts of a microscope. You'll be having a quiz next week in which you're going to have to know these parts, identify them, and tell what they do.
next week.
So, the compound microscope has two lenses that magnify the image. One is here, and the other are these lenses that you sort of can rotate through. So number one, the one you look through, it's called the eyepiece. And it has a lens in it that gives a certain amount of magnification. This eyepiece in our microscopes gives a 10x magnification.
doesn't seem like enough.
The other lenses here are called objective lenses. And you can rotate our lenses. We have 3 objective lenses on our microscopes. Low, medium, and high power, we call them. Our microscopes go from low power is 4x, medium is 10x, and high power is 40x. Some microscopes might have a 100x objective, but oftentimes it requires oil in order to use it. It's called an oil immersion lens. The part you hold on to and you sort of rotate these objective lenses is called the nose piece. And as you're using these microscopes, it's important that you always, so when I'm changing from one power to another, it's important that the objective lens gets clicked into place. If I rotate it like halfway, you're not going to see anything because it's not in the right position. So you got to make sure it clicks into place. You'll feel it once you use it. Colin?
If I were to want to acquire one of these, how much would it cost?
So like a microscope like this is probably about, it depends how you buy them, probably about $400 to $500.
Oh, that's it?
Well,
So, you know,
how many do you have?
So sometimes, all right, listen. Sometimes you can go like to Walmart and Target, and they do sell like microscopes. They're typically like made of plastic. The lenses are not good quality. Sometimes those kind of microscopes, you might be able to see something under low power, but really the high, and sometimes they'll say magnifies 1,000 times. They will never do that. So you can buy sort of less expensive ones. You can buy toy ones, but you know these are relatively inexpensive as far as microscopes go. You could buy a microscope that costs $1,000, $1,500 easily.
But it's also free if you steal one.
Ollie,
2 questions. I'll start with the first one. You never actually said how much the electron.
Yeah, that's what I was.
how much they cost?
Yeah.
Yeah, they're not like, they used to be super expensive. I would guess you could buy them used for $10,000 to $15,000 maybe.
You
I'm sort of guessing here.
I'd say.
I'll look it up for you guys at some point. All right, we need to move on though.
body tube.
Ollie, it's quick.
Okay, when are we gonna actually use microscopes?
Probably tomorrow. All right, so
Wait, are we gonna dissect?
this part between the objective lens and the actual eyepiece lens is called the body tube. The lenses need to be the proper distance apart in order to focus the image. On the bottom is the light source. It can vary. On our microscopes, we have cordless, it's battery powered. You can have LED lights, you could have fluorescent lights. Sometimes microscopes have to be plugged in. This microscope, this is like an old school microscope. I wonder if I have any of these left. But it has not a light source there, it has a mirror. And you had a separate light source you plugged in, you aimed the light at the mirror, which reflected it up through the objective lens. So this is kind of an old style of microscope. The place you put your slide that you're going to view is called the stage. And then on most of our microscopes, there's these little clips that hold your slide in place. They're called stage clips. But I do have one microscope. It's a little bit nicer microscope that has something a little different. And it's called the mechanical stage.
Oh, I'm so excited.
And the mechanical stage is pretty cool.
It's dissecting.
So the mechanical stage, rather than just clips,
So
has this little thing here. And then, because on those microscopes, in order to move the slide and look at different parts of it, you basically just move the slide with your fingers. You know, like this. And sometimes, if you're under high power especially, you try to move it a little, but it goes flying off, you can't see anything. This microscope has these little knobs here. And what they do is they move the stage forward and backward. They move the slide left and right. So you can get very precise control over how the slide's moving. So it's a little bit different type of microscope. When you're looking at a microscope, the amount of light that you're allowing in is really important in what you see. Sometimes you need it brighter. Sometimes you need it darker. And so on the microscopes underneath the stage, there's a little mechanical piece called the diaphragm. It opens up this hole larger or smaller to change how much light is going through and reaching your eye. Sometimes you look at these microscopes and it's like blindingly bright. It means you need to close down the diaphragm so it's smaller and less light's getting through. Other times it's too dark and you open up the diaphragm to see more light. The bottom of the microscope is called the base. Colin, let's worry about our nose. The part that you sort of I'm holding on to right now is called the arm of the microscope. And then the most important parts, or two of the most important parts, are these knobs here. And people often mistake what they're for. So you can see when I turn this knob, what's moving?
Oh, it's a stage.
The stage is moving up or down. Same thing here. It moves the stage up or down. And moving the stage up or down doesn't do what a lot of people think it does. People believe that like zooms in on the image. It doesn't. Changing where the stage is changes the focus, how clear an image is. This stage needs to be at the exact right spot in order to form a clear image in the eyepiece. And so the knob that moves the stage up and down helps to focus the image. It's called the coarse adjustment because it moves a lot. Like when I turn that, you could see the stage is obviously moving. There's another knob though, a smaller one. When I turn it, you can't see anything moving, but there is something moving. Okay, this is called the fine adjustment. It's moving the stage as well, but it's moving it just a tiny amount. Because when you're in high power, magnifying an image to a great degree, you only need to move that stage up and down less than a millimeter. So that fine adjustment allows you to do that. And those are the parts of the microscope. The dissecting microscope just has one focusing knob, that's this, and it moves this up and down. The other thing it has is this knob, which you can move the whole apparatus up and down. Usually you want it about halfway.
Whoa.
All right, I'm going to skip this because we just did it.
About
Yes, Lucia.
why are they all named after like parts of the body and stuff?
I don't know, that's interesting.
The eye.
All right, let's have a little, let's see if we can identify some things.
I swear.
The diaphragm.
Anyone, raise your hand if you want to take a guess. So these are all
Oh, I think I got it.
images taken under a microscope. They're everyday objects that you're probably familiar with, although you might not be able to recognize them. Ali, what do you think it is?
Okay, I think it's a wristband under an arm.
No?
No, I got it. It's a fork.
No?
Dang it.
This is?
Water? It looks like someone is behind bars, and that's complex.
So,
What?
this is actually a CD or a DVD.
What?
These are the rings around the outside of the DVD that your DVD player reads data from. All right. Raise your hand. I'll take one guess for each one, otherwise we're going to be here all day. Raise your hand. Any idea? So it's magnified quite a bit. So it's not like what it looks like. Yeah, it's not steps. This part will give you a tip.
Oh, I think I got it. Is it like
No?
toothbrush?
No, you're close. Another hygienic tool.
Hairbrush?
No. I haven't used one in a while.
Floss.
Hairbrush.
No.
Shaver.
Yes, it's a razor.
That's
These are the blades. You could see the sharpened edge. These are little hairs
See, that didn't say
on
hair.
them.
That is not
No, it's not Cocoa Krispies. It's not cereal.
Hot chocolate.
If it's something that's not
It's magnified. No.
I have no idea what this is.
These would be like microscopic marshmallows.
Crystallized. Is it gravel?
tofu.
No, it is something you eat. It almost is gravel.
Sand? Rice?
Minerals.
Something.
Salt and pepper.
What?
That's cereal.
Those are whole marshmallows.
These are salt crystals. These are flakes of pepper.
Those are marshmallows.
It's salt and pepper.
I know the next one. I know the next one.
Not salt and pepper. All right.
We all know this one.
This is easy.
I don't.
Yes, Lily?
Velcro.
Velcro.
Yeah.
I knew that one.
This is,
color.
when you think about Velcro, the like scratchy side is this red side. Velcro is made out of little plastic hooks on one side, and then the soft side of Velcro is just sort of wound up fiber. And when you push it together, the hooks latch on to the loose thread, and then when you pull it, these are flexible and they just kind of detach. That's how Velcro works. We have more, don't worry.
Yay.
That is like
What kind of paper?
What is it?
Toilet
I knew it
paper.
was pizza.
Wait, what?
Now this, I don't, you may not know.
Slinky.
No.
Rope.
Rubber band.
So I don't know if people really know what this is. These are nylon stockings, you know.
Oh, we literally.
These are nylon fibers that are wound into
Is
that
that in
shape.
my home and career school?
What is this?
The earth.
It is not an eye. It's something very small.
A marble.
It's the earth.
That was not a marble.
It
It's a drop
is.
of water.
Many of you are holding one right now.
Water.
I'm not holding anything.
It's a water. I'm holding
Not a pencil.
paper.
It's a ballpoint pen. This is the ball that rolls around, and that's how the ballpoint pen works.
They chew that.
The great divide. That's the Grand Canyon. That's the Grand.
This is a record. The grooves on a record.
Oh.
I know what they're Dorothy
Not French fries.
Poppy sticks.
These are small things.
Cigarette. What magic?
Toothbrush.
Toothbrush bristles.
Oh, I got that one.
Really?
Plant.
Hair.
It's not what it looks like.
It's grass.
Those are eyelashes with makeup.
Oh,
Makeup is not
Makeup's
green.
on.
I'm sorry.
I've seen this image before.
That's hair. That's hair.
This is what comes
That
up
is
with
That
hair.
is dental floss with tooth gunk on it.
Tooth gunk is not pink.
I eat pink stuff.
All right.
Don't tell me that's pink.
So tomorrow we will start to use and practice our use of the microscope. After we talk about the steps to do that.